Welcome to my video about DNA mutations, specifically point mutations. Here we have our original DNA strand, and we need to find where we can form a 5 amino acid protein after transcription and translation. Searching the two strands, we find this start sequence and this stop sequence, keeping in mind that the T's will become U's after transcription. So the bottom strand is the coding strand and the top strand is the template strand. Here, the coding strand has just been turned around, so we can work with it 5' to 3' left to right for the rest of the video. The start and stop sequences have also been color-coded. After transcription, the mRNA strand will have the same sequence as the coding DNA strand, except the T's are replaced with the U's. Each codon has also been color-coded, so we can see the 5 amino acids with the stop codon at the end. Using the amino acid chart, these are the amino acids that are formed after translation. The UAA at the end doesn't code for an amino acid. Now let's go through 4 different types of point mutations, but before that we will review transitions and transversions. Here are the 4 nucleotides. T and C are pyrimidines and G and A are purines. Point mutations result in the substitution of one base pair for another. A transition is a switching of a purine to a purine or a pyrimidine to a pyrimidine. A transversion is a switching of a purine to a pyrimidine or a pyrimidine to a purine. Here is the original DNA coding strand. Here is the mutated DNA coding strand with the mutation being pointed at. Because a purine was changed to a purine, this is a transition. Here are the original and mutated mRNA strands after transcription, and here are the original and mutated amino acids after translation. Because the mutated codon ended up coding for the same amino acid, this is a silent mutation. Imagine you needed green paint but picked up someone else's order at the store. However, that other bucket ended up being green too. Next, we have this mutation in the DNA, resulting in this mutation in the RNA. Because a pyrimidine was changed to a pyrimidine, this is also a transition. Looking at the amino acids from the original and mutated mRNA strands, we see that this is a missense mutation, since the mutated codon coded for a different amino acid. When you picked up the wrong bucket at the store, you got red paint instead of green. In this third mutation, we have this A changed to a T. Because a purine was changed to a pyrimidine, this is a transversion. After translation, we see that the mutation has caused a codon that is supposed to code for an amino acid to become a sequence that signals the stop of translation. This is known as a nonsense mutation. Because you ran out of paint, your wall was not painted completely. Lastly, we have a mutation in the codon that signals translation to stop. Because a purine was changed to a pyrimidine, this is also a transversion. Let's go through translation so we can see the impact this mutation has on the protein. Here we can see that where translation was supposed to end, the mutated codon coded for an amino acid, and translation continued to occur, resulting in a longer than normal protein. This is a read-through mutation. After painting was completed, many buckets of paint were still left over. Here is a summary of the 4 point mutations that were discussed in this video, and an example of each that we saw.